Start the recording. Yep, and you did. Okay, and we have some additional people coming in. Presenters, can you see that the participant numbers have gone up? Can you see that as well? Great. Okay. Welcome everyone. We'll get started in just a moment. Not even a minute. Okay, marvelous. All right, everyone. Welcome to Body Functions, and this is an interactive poster session. We are going to start with Michelle Hernandez, pronouns she, her, hers. She is a senior. She's a biology and human physiology major with a minor in chemistry. And they're going to be talking about the poster today, inflammatory biomarkers concurrent with the progression of osteoarthritis. And their research mentors are Angela Lina Castro, Kelly Languici, and the principal investigator is Bob Goldberg, or Robert Goldberg, and those are all with the Knight campus. And she is also an executive board member of the Minority Association for Pre-Medical Students, MAPS, and a biology undergraduate teaching assistant, which is a wonderful way for them to share their knowledge with other undergraduates who are going through the classes that they did well in. So if you'll take it away, it is all yours.
Thanks, Annie. Okay, so can you see my screen? Good. Okay, cool. So hello, everyone. My name is Michelle, and today we are going to be going through my poster on characterizing inflammatory biomarkers concurrent with the progression of an injury-induced model of osteoarthritis. So osteoarthritis is a chronic and progressive degenerative joint disease characterized by the progressive loss of articular cartilage, changes in subchondral bone, and inflammation of the synovium tissue. It affects nearly 33 million people in the US and is one of the leading causes of chronic disability. Currently, there are no therapeutics or disease-modifying OA drugs available for this disease, only ones to help manage the pain. However, when the symptom management is no longer a viable option in the later stages of the disease, a full joint replacement surgery is the next suitable option, though it's very costly. About a year ago, our lab conducted a pilot study that set the stage for the two ongoing studies that we have going today. This 3-week study, which I will be discussing today, and the longer 6-week study that is also ongoing. In the pilot, we demonstrated the feasibility of longitudinally monitoring immune responses in an injury model of OA. We were able to see some elevated biomarkers in the injured groups when compared to the controls. However, nothing was deemed significant. With this study, we introduced a sex variable as well as a therapeutic to analyze how these factors could be impacting early biomarkers in an injury model of OA. The main focus of our research is to understand the immune response concurrent with the progression of the disease. The overall goal with this research is to identify early immune biomarkers that may be indicative of the disease status. As you can see down here, we used a well-established model known as the medial meniscal transection model, or MMT, in the rat. For this, an incision is made in the medial side of the knee with the goal of exposing the medial meniscus. The meniscus is then cut, and this simulates a complete tear. Due to its poor vascularization and low cellular density, cartilage has a limited regeneration turnover. So by cutting the meniscus, we create a mechanical instability that does not get repaired, and it leads to joint destabilization and intra-articular inflammation. For this study, we had an N of 32 and 6 groups. So we had 6 with the MMT surgery and a therapeutic, 6 with the MMT no therapeutic, and 4 shams, or 4 controls. And this was the same among the males and the females. In order to quantify the changes that occurred due to the disease, we used a contrast-enhanced microcomputed tomography, or a micro-CT method. After the micro-CT images were taken, we ended up with these 2D grayscale images, which then can be turned into 3D representations of tissue and bone. With these analyses, we measured quantitative OA metrics such as cartilage thickness and volume. In order to quantify the immune response in the animal model, we used longitudinal blood draws and an endpoint synovial fluid aspiration. We conducted weekly blood draws for the duration of the study, so for 3 weeks, and we had an endpoint synovial fluid aspiration. In order to identify the circulating immune cells, such as T cells, macrophages, and B cells, we utilized flow cytometry. And in order to measure the circulating inflammatory cytokine levels in both the blood serum and the synovial fluid, we will be using a Luminex multiplex assay. And so, as I mentioned before, this study is ongoing. So while we have not had the time to analyze all of our samples, we have run the micro-CT evaluations on a few thus far, which is depicted in the upper right-hand corner here. So here we have the data normalized to starting weight. We normalize the data here to starting weight because of the fact that we're working with males and females, which differ in weights pretty significantly. And weight has been shown to have an influence on how much damage can occur in osteoarthritis. This is why the weight loss is one of the first methods of treatments that doctors prescribe to patients with the disease. And so far here, we see possible sex differences with the OA treatment. As you can see here, the females, regardless of the intervention, there does not seem to be a big difference in the cartilage thickness. However, with the males, you can see that the group that received the therapeutic had a much more thickness than the ones who did not receive a therapeutic. This is also visible in the cartilage thickness heat maps of the proximal tibia to the right. The cartilage is colored to indicate cartilage thickness, so the red indicating thicker cartilage than the green. Notable is this male, which received no therapeutic and has lots of damage and the lowest thickness point on the graph. Here we see some pits and some divots and lesions, which represent heavy damage. These results suggest that there could be a possible sex difference in how these animals are responding to treatment. This will be interesting to investigate further once we continue to analyze more samples to see if these differences are real. And it'll be exciting to see whether these are correlated to differences in immune biomarkers among the two sexes. So for our next steps in the study, we are going to take all of our 2D grayscale images and calculate the surface roughness of the cartilage on each sample and compile that data with the other quantitative OA metrics we have. From there, we plan to employ regression techniques to identify immune biomarkers that are correlated with the OA metrics, such as the surface roughness of the articular cartilage at the endpoint. Thank you for listening.
That was marvelous. We have time for a few questions, and then we'll also have time at the end of the session for questions for everyone, or if you need a little bit of time to just let the wonderful material sink in and so you can formulate a question. I didn't see anything. Oh, Kelly says, great job. I agree. How about, what about something that surprised you about this research?
Something that was pretty surprising to me when I first started getting into this research, which was how much it affects like the US population. I had never heard of the disease prior to coming into my research lab. So it's pretty shocking to learn that 33 million people suffer from it.
Oh, fantastic. While folks are thinking of other questions, I have a follow-up, which is, what's the experiment that you're hoping to do next? Or if you don't have one sort of in line, like what's an experiment that you would want to do next that may or may not be even physically possible?
Yes. So in, I guess, finishing up this study, another possible way to go would be to add exercise therapeutic, because exercise is also another frequently prescribed method of intervening for osteoarthritis. So having that as a variable to see how that influences the disease would be interesting.
No, cool. All right, looks like we have a couple of questions now. I'm going to read them out loud. Dr. Walker asked, what are your expectations for the difference in inflammation between males and females?
Yeah, that's a great question. I would suspect to see more inflammation in males just because of the possibility due to the different weights, but I'm not entirely sure yet. So I'm interested to see what we find.
That's great. Okay, do you see the other question?
From
Dr.
just down here.
Dr. Heterachi. Great presentation, again, absolutely true. Has anyone done similar studies in the past looking at immune cells and cytokines in OA that might help inform your hypothesis for your own results?
So yeah, people have looked at inflammatory cytokines, and there are established ones, such as like IL-6 and TNF alpha. And so we know those are a sign of the disease, but we were hoping to look into ones that are kind of a disease indicator, I guess you could say. So tracking those in a longitudinal manner is what kind of we're looking into.
Great. Thank you so much. Okay, I think we can let you take a break. A round of applause. Nice job. Okay, terrific. So as we prepare for the next presenter, Ireland Johnson. And really terrific job. Thank you so much. Okay, so Ireland is a junior. Her majors are biology. Her major is biology. Her minor is chemistry and Spanish. She's going to be presenting on design and cytocompatibility of hyaluronic acid hydrogels for bone regeneration. And her advisors are Andres Guerrero, Veronica Spalding, and Dr. Marian Hederachi. Okay, take it away, Ireland.
Awesome, thank you.
Thank you.
Just share. Awesome, so hi, just to reiterate. Yeah, is there, okay. So hi everyone, just to reiterate, my name is Ireland Johnson, and I'm an undergraduate from the Hederachi lab at Knight campus. And today I'm presenting on the design and cytocompatibility of hyaluronic acid hydrogels for bone regeneration. So large bone defects and fractures caused by trauma or disease remain a serious challenge for orthopedic surgeons. And therefore there's a need for more effective treatment strategies to repair injured bone. So bone allografts, which are grafts from a donor of the same species, are often used to heal large bone defects, but have a high chance of immune rejection and often fail to integrate with the host bone, mostly due to sterilization processes that remove viable cells and proteins. Autografts, which are a tissue graft from the same patient, are ideal because there's low host rejection and the graft is not weakened from sterilization. However, they're not widely available and can cause donor site morbidity. So to combat this problem, biomaterials composed of natural polymers like collagen or hyaluronic acid can be used to deliver bone-forming proteins that produce an osteogenic healing response. So more specifically, the objective of this study was to develop, design, and test the cytocompatibility of hyaluronic acid, abbreviated HA-based hydrogels for bone regeneration applications. So HA hydrogels were formed via dynamic covalent bonds between varying types of gels containing aldehyde or hydrazide functional groups at 2.5% weight per volume. We tested 5 treatments. The first oxidized with carbohydrazide HA, second pentadiol oxidized with carbohydrazide HA, third oxidized with adipic acid dihydrazide HA, fourth pentadiol oxidized with adipic acid dihydrazide HA, and fifth was a tissue culture plastic, which served as a positive control. So each HA hydrogel combination was seeded with 3T3 fibroblast cells, which is visible in figure 1. And cell growth and viability, which is seen in figure 3, were measured at 4 different time points, day 0, 1, 3, and 5. Live cells were evaluated using green fluorescence from GFP, and dead cells were evaluated using red fluorescence from Ethidium homodimer. Fibroblast cell culture images 5 days post-treatment are observed in figure 2. There was no statistically significant difference between any of the treatments in regards to cell viability over time, which is noted in figure 3A. However, there was a trend indicating that apart from the pentadiol oxidized with adipic acid dihydrazide HA, treatments in all other conditions maintained high comparable cell viabilities over time. The most noticeable difference in trends were observed when looking at cell count over time, which is noted in figure 3B. There is a statistically significant difference between the oxidized with carbohydrazide HA and the pentadiol oxidized with adipic dihydrazide HA on days 3 and 5, as well as a statistically significant difference between both pentadiol oxidized HA treatments on day 3. Additionally, there was a trend indicating that oxidized with carbohydrazide HA showed high cell viability and a rate of cell growth that surpassed all other conditions. For all 4 time points, this particular condition maintained cell viability greater than 82.3%, and a cell count increase over the duration of the study of 554.2%. And so since this particular treatment group of oxidized and carbohydrazide HA performed well, we're currently determining if different ratios of oxidized or carbohydrazide produce varying rates of cell viability or cell count. And so a future expansion of this project would be to determine if these hydrogels could become protein delivery vehicles of osteogenic healing proteins, such as bone morphogenic protein 2, which could ultimately lead to the use of HA hydrogels as biomaterials that rival the healing response of bone autografts. I'd like to thank the members of the Hedy Lab and Knight Campus for assisting with this project. And this project was also supported by the Peter O'Day Fellowship, as well as the Knight Campus Undergraduate Scholars Program. So thank you, and I guess we're opening the floor to questions now. So if anyone has any.
As terrifying as that is, yes. Wonderful job, Ireland. Thank you so much for sharing this amazing work.
Yeah.
And your fellow panelists can also ask questions as well if they'd like. So you talked about your future work. Was there anything that was particularly fun about what you learned here, or what was the most interesting thing to learn?
I really like this project is very closely in line with my career goals of like becoming like a surgeon scientist. And so being able to like see how like using biomaterials could eventually someday be used clinically was a very exciting prospect for me. Yeah, I hope that answers your question.
No, it's great. It's awesome. What a great experience too.
Yeah.
Was there anything in particular that inspired this work?
Our lab focuses a lot on biomaterials and tissue biomedical engineering. And I tend to do a lot of cytocompatibility tests within my lab, and I guess it just kind of fell into place. Yeah.
That's great. Okay, are there other questions from the audience or the other panelists? Okay, folks might be thinking about questions to ask at the end of the session, so we'll feel free to put them in the Q&A part, and we'll keep track of them for Ireland for the future as well. Okay. Okay. Next up, we have Sahana, Krishna, Kumaran, and hold on one second. I'm sorry, I was on the wrong screen. They're going to be talking about old Elastin, haploinsufficient mice that have impaired memory, motor coordination, and endothelial function. And are you able to?
Yes, I will share my screen with everybody.
Great. That moment where we wait for it to see if it's working. It looks great. Even better. Looks perfect.
One moment.
These are things that maybe you'll have to think about in the future, but for now.
Okay, so thank you for the introduction. I will be talking about elastin haploinsufficiency and effects that it has. So with aging, our large elastic arteries like the aorta and the carotid artery will get stiffer, in part due to the breakdown of elastin. So elastin is a protein that makes our arteries elastic, basically stretchy, and a stiff artery is kind of like a pipe. It's not elastic, so it can't expand enough to accommodate every pulse of blood coming from the heart. Therefore, it can't dampen the pulse of blood, and that leads to bigger spikes in pressure in smaller vessels, like the cerebral arteries. And these arteries are not structure to be exposed to high pulse pressure. So previous studies with young mice with large artery stiffness have shown that these mice have decreased cerebrovascular function. And there are multiple other studies that highlight the correlation between large artery stiffness and cognitive decline. However, the direct effect of large artery stiffness and age together has not been completely studied. So this study looks at old elastin haploinsufficient mice, which I'll be referring to as old ELN plus minus mice. They only have one copy of the elastin gene, and therefore they have large artery stiffness. And we compare them to old wild type mice, referred to as old ELN plus plus mice, and young wild type mice, young ELN plus plus mice. So our hypothesis is that old ELN plus minus mice will have decreased cerebrovascular function and cognitive function compared to both of the wild type groups. So to test this hypothesis, we first ran an accelerating rotarod test, a test of motor coordination. So as you can see in this figure, we found that old ELN plus minus mice have impaired motor coordination compared to both of the wild type groups because they spent less time on the rotarod. So we also ran the Morris-Water maze test, which is a test for spatial memory. So in this test, we fill a tank full of water, and there's a platform in the tank. So mice don't like water, they want to escape the water, so they should be incentivized to remember where the platform is. So in this figure, you can see that the old ELN plus minus mice have impaired spatial memory because they had a higher latency to platform area, basically meaning they took longer to reach the platform area the first time. And they also crossed the platform area less frequently compared to the wild type groups. And you can see that in the representative track path images as well. So these results put together indicate that old ELN plus minus mice have impaired cognitive function. So we also assessed cerebrovascular endothelial function, and to do so, we studied ex vivo pressurized posterior cerebral arteries. So this figure is showing the results of those experiments, and we found that old ELN plus minus mice had significantly lower endothelium-dependent dilation compared to old ELN plus plus mice, as you see in part A. And the old ELN plus plus mice also had, in turn, lower vasodilation compared to the young control mice. But when we added L-name, a molecule that inhibits the production of the vasodilator nitric oxide, the maximum vasodilation in all groups decreased. And this indicates that in old elastin haploinsufficient mice, impaired endothelial function is due to the lower bioavailability of nitric oxide. And we know that the impairment is at the level of the endothelium because in figure 3C on the right, which shows endothelium independent dilation, there are no significant differences between groups. So therefore, the impairment is at the level of the endothelium. So finally, we assessed neuroinflammation in the entorhinal cortex of the brain. The entorhinal cortex works with the hippocampus and functions in memory pathways. And we studied neuroinflammation by measuring the abundance of microglia, which are immune cells that cause inflammation. So we did so by analyzing the percent of the entorhinal cortex that's positive for IBA1, a marker for microglia. So you can see in figure 4 that we found that old ELN haploinsufficient mice have a higher percentage of the entorhinal cortex taken up by IBA1 compared to the old ELN plus plus mice, indicating more microglia. And you can see in the images on the right, the IBA1 stain shows up as a brown color. So you can see in the bottom image, one from a old elastin haploinsufficient mice, there are more brown spots indicating more microglia. So in conclusion, old elastin haploinsufficient mice have impaired cognitive and cerebrovascular function, basically indicating that large artery stiffness caused by aging plays an important role to changes like other organs in the brain. So I just want to thank the Office of the Vice President for Research and Innovation, as well as the Alzheimer's Association for funding this research. And if anyone has any questions, I would be happy to answer them.
Marvelous work. Thank you so much. How did you come up with this topic to study?
Yeah, so there's a lot of research showing that in Alzheimer's disease, like the vascular system, especially the aging vascular system, plays an important role in the effects that occur in the brain. So studying something like the cerebral arteries, basically blood flow to the brain, can really impact a person's cognitive ability, which is why we took it one step further and we look at large artery stiffness, something that occurs with aging, and just seeing the downstream effects of that and how that affects blood flow in the brain.
That's awesome. Thank you. Very cool. What about what would be your next experiment? Or, and it can be like what you would actually do, or like what's the like crazy experiment that you'd want to do if it was.
Yeah, so something I think would be very interesting is looking at inflammation in the cerebral arteries themselves. Just trying to look at how endothelial dysfunction is tied into inflammation and how that leads to inflammation in the brain. So just focusing a little bit more on the cerebral arteries and trying to see like what mechanisms are at play.
Very cool. Okay. Do we have any questions from our attendees? Okay, maybe folks are thinking about questions for the end. Thank you so much. What a wonderful presentation. So last and definitely not least, we have Albert Yim. He is a junior and he's majoring in human physiology and neuroscience. I'm going to be talking about wearable biosensors, and he's with the Reader Lab.
All right, hi everybody. Yeah, so my name is Albert Yim, and I'm part of the Reader Lab, mentored by Dr. Jonathan Reeder. And my project is about wearable microfluidic colorimetric sweat sensors for the real-time personalized hydration monitoring. So, little introduction is sweat is a valuable resource that's not fully understood yet. Sweat rate and composition are highly variant between individuals. So that poses the question on how we can develop a system for personalized hydration feedback. Fluid loss is critical in knowledge about proper hydration, and biochemicals in the sweat can be indicative of physical exertion. There's also possible clinical uses for this, as cystic fibrosis is identified by elevated electrolyte levels. So sweat biomarker variance is very attributed to sweat rate. So this poses the challenge of recording a continuous sweat rate to give our biomarkers practical use. So the approach for this project, we use our microfluidic devices. These are made from silicone elastomers, and these will help us collect and store sweat non-invasively on the skin. So sweat enters the inlet hole and it is stored through the channels in the device. And these channels are sealed by the two bound silicone layers. There's an outlet hole in the silicone layer that allows for proper flow and is engineered to prevent backflow and contamination. The color you choose will try to dissolve dependent on flow rate. That the color development through the channels will be dependent on flow rate. Studying this color development will be the basis for our colorimetric system, where we can visually analyze unknown sweat rates in collected sweat. So in order to test that our dyes are flow rate dependent, I'm using an experimental setup that uses pressure at set physiological flow rates, and taking an image with the camera, we can process this image in Photoshop, where we can correct for white balance and record CIE LAB lab values. So statistical analysis of each outer turn or serpentine will determine if there's a color difference between different flow rates. So far, we've tested the physiological flow rates of 1 microliter per minute and 0.5 microliters per minute. So analysis so far has found a difference in the 5th through 10th filled serpentine. And right now we are trying to change the experimental setup to try and combat like the noise and variability in our experiment. So right now, if you could see in the two images in the upper right-hand corner, like right now, the whites are a little bit different and the lighting is inconsistent. So in order to combat that in the future, we are going to start shooting with a camera opposed to a phone, so we can record in RAW opposed to JPEG. This means that the images are going to be less processed and we can manually process it ourselves more in Adobe Photoshop. And then to combat the light inconsistencies, we've removed the light source. So right now it's only indirect light. So us being able to find a difference in color development will allow us to study the relationship between specific flow rates and specific color values. I'd like to thank everybody else in the Reader Lab for helping me in my project.
Thank you so much. So I've been trying to figure out, like, so is this going to be something that we wear, like, is it going to be a whole shirt of this thing, or is it going to be like a little patch on a shirt that we would wear?
I guess a little scale would be helpful. This is like probably about like this big. So you could fit it like right now where we study it would be on your forearm, like right below the elbow. So it wouldn't be that big. It would just be, yeah, a way to non-invasively measure your hydration levels.
Okay, cool. Neat. Thank you. Sorry, I couldn't quite picture it. So, I'm fascinated how you came up with this idea. How did this come to pass?
I personally didn't really come up with this idea. This was Dr. Jonathan Reeder's kind of idea. He came from a lab recently at Northwestern where they're doing very similar work, and I just came across it. I thought it was just super interesting. So like it kind of applies like because I'm really interested in like training and I guess monitoring like performance metrics for like athletic performance.
Yeah. Very cool. All right, so the question I've asked all the other panelists. So what's next?
So yeah, next is like just to kind of keep on adjusting our experimental setup. That way, like we have no outside variables, and so we can actually determine if the dye that we are currently using is flow rate dependent or not. And if we can figure out that if it is, then we can try to like use that development to like reverse, like find out a sweat rate. So like we can go out into like into the real world and collect someone's sweat and then analyze their sweat rate to give them personalized hydration feedback.
Okay. Very cool. I'll look forward to that.
Thank you.
Thank you so much. What a wonderful set of presentations. We don't have any questions from other attendees at the moment. Do any of the panelists have questions for each other? I'll also tell you that if we were doing a live poster session, there would be plenty of awkward standing around there too. Thank you to our attendees who are here. Thank you to the presenters. What amazing work that you're able to do. Is anyone going to be working on research for the summer? Terrific. And are you going to be continuing some of the same work, or are you going to be heading in new directions?
For me personally, I'm going to finish up the work that I'm currently working on.
Great, fantastic, good. I wasn't thinking of additional questions, so as much as anyone who knows me is going to be surprised for me to hear me say this, but I don't have any additional questions to ask at this time. Did anyone think of, any of the panelists think of questions to ask each other? Okay, how about one more? For everyone on the panel, if you could give somebody advice about how to do work like this in your lab, what advice would you give them? Or if you want to think about it, like, what's the advice you would give your younger self, so that you could get to this, get to today? So advice to somebody who wants to do this work in the future, or advice you would have given yourself a year ago.
I can go first. That it's totally okay to have no idea what you're doing. And
That's
it's
awesome.
okay to stumble through it and ask for help, because everyone's been there at some point. And you learn and you pick it up pretty quickly once you get going. But yeah, I was definitely clueless for a long time until I figured it out. But yeah.
That's awesome. Thank you. A lot of adulting is stumbling around. Sorry, Sahana.
I was just going to say that, like, science is imperfect. You're not going to get those, you're not going to always get the results that you expect. And part of that is just the nature of science, of course. So you just need to take things as they are. And I think also just like keeping those expectations in mind that things are not as like perfect as a textbook makes them look like.
Yeah, we sort of do a disservice, right, by showing you the things that all worked out and we figured out, when so much of science is like, what are we going to do next?
I can go next. Kind of to build upon what Ireland said, to not be afraid to ask questions and ask for help, and also not being afraid to like keep pursuing or being persistent on trying to find a research lab, because I know it's not always the easiest thing to come by. So definitely, like, if you want it, you can try to find somewhere that will take you, and you start out probably really small doing basic things, and then you build upon that and you get to work on a project like everyone else on the panel. So yeah, just be persistent.
That's awesome. What do you think is like the best advice you could offer for being able to get into a lab, to get to do a project like this?
I think just, I guess, depending on your discipline. So if you're biology, like, we have resources where we have all of the biology labs listed, and we usually tell incoming freshmen, like, here are all our research labs. Don't hesitate to reach out and see if anyone's looking for undergraduate researchers, because as you know, like, we're only here for 4 years, so graduate, move on, and they're always looking for more people. So I guess cold emailing, cold calling is probably the best way. And there's also programs like, I think Ireland said she was in it, the Night Campus Undergraduate Scholars. I was in it last year. So programs like that are also really great because they pair you with a lab.
That's awesome. Thank you. Yeah, I know one faculty member said that he might ignore an email, but if you came to their office hours or something, then they're more likely to figure out how to work with you. So after all that work we've done to get you to send emails, also show up in person. Okay. Albert, do you have any advice for your younger self or for others who are interested in doing the work that you're doing?
Yeah, I would just say, like, it takes, like, at least for me, research takes like a lot longer than I thought. And definitely use like your resources in lab. Like, you can learn something from like every single person in lab. Just pick up like their best habits. And yeah, that's that's what I got.
That's great advice. Yeah. Awesome. Yeah, that's something I know that when there's a librarian project and we interviewed recent graduates and the folks who hired them, and they said that they're also employers were surprised that students didn't ask the people who worked in the offices of the labs where they were hired for information, that they thought everything was going to be all online, and so that they were looking forward to interacting with the new folks and to sharing the information that they had as well. So encourage you to do just more of that. That's awesome. Okay, did anyone think of a question they wanted to ask?
I guess I can just ask like a general panelist question, like what are the end goals with all of your guys' project? Like, you know, the big picture of if in the future, if everything goes to plan, like what do you hope comes out of the research?
For my own big picture would be a clinical study. Slightly smaller picture would be like a rat model. But there's a long way to go until that point, but yeah.
I guess for my
You can go first.
thank you. I guess for my project would just be like, yeah, being able to move on to like trying to identify like more like complex biomarkers, like maybe like lactate, like glucose, just more things that would be indicative of like more like physiological performance and like diseases, not just hydration.
And I guess to answer my own question, I can go next. So I think our overall hope with the research is like one day to be, if we find biomarkers that are concurrent with the disease status of osteoarthritis, to just take a simple blood test after someone has a severe knee injury to know the disease status, so you can like intervene in the earlier stages and prevent any further degeneration, rather than finding and discovering the disease once it's manifested and disability has occurred.
For my study, I guess the end goal would just be to show that link between the direct causal link between large artery stiffness and cognitive decline that leads into dementia. And I guess going on from that, it would be great to look at potential therapeutics for large artery stiffness and see if those work to reverse the effects of dementia.
Oh, great question and great answers. Nice work, everyone. Okay, any additional questions? Okay, it is 2:30, so you have a little bit of time that you might have allocated for this panel, but I think we can safely say that we've had a wonderful exchange of ideas. Thank you so much for enlightening me on the work that you're doing. It is so awesome to get to know a little bit more about the work that you're doing in your labs and to know a little bit more about the labs. And I wish you all the very best success. I hope everything goes well this summer and in the coming year. Is there anything else? And also, let me thank also Ellen Liang. Sorry, I didn't even ask how to say your name, for being the co-moderator on this. I really appreciate everything. And we'll end the recording and send you all on your way.
Thank you, Annie, and thank you, Ellen.
Thank you.
Thanks, Michelle. Thanks, Ireland, Sahana, Albert.